Confirming again, can everybody listen to us? Yeah, I guess.
Yes, yes.
Perfect. So moving on. Okay, perfect. Okay. And let's get started because Professor Turakhia has a very tight agenda today. Okay. So it is my great pleasure today to have Yatish here. We know him for very long for his work, but also because we have interacted quite a lot. I don't know how to summarize his very successful career, but I'll do my best. He's currently assistant professor in the Department of Electrical and Computer Engineering in UC San Diego, which is a very good position, I might say. But he has a very long track on working in this type of topics. He did his PhD at Stanford University, and he did his postdoc at the UC Santa Cruz. During his PhD, he was advised no less by Bill Daly, which, if you remember, he's currently the chief research at NVIDIA. So he knows a lot of tricks on this. He has very important contributions to science and to research, but I would like to highlight two of which I like very much. One of them is a living proof that the things we can get to do, they have a real impact on society. I'm talking about his Nature Genetics Usher, which is a tool that enables a rapid placement alignment of sequences of the SARS-CoV against phylogenetic trees. And this is a key and useful tool for real tracking of outbreaks like the one of the COVID. So this proves that the algorithms and accelerators we do, they do really have an impact on society. The other piece of work that I've chosen, and I believe it represents very well what he does and his expertise, is the Darwin Accelerator. Most of you have read the paper, have worked understanding the Darwin and trying to improve the Darwin, I might say. And he's a very good example of hardware-software co-design. He developed this, and it's a very, very, very nice piece of work that I recommend everybody to read. Today, we have the pleasure and the honor of having Yatish here. He's going to give us a talk on algorithms, software, and hardware accelerator for the next wave of genomic data, which is pretty much aligned with all the things we do here at the group. I'm sure you guys will enjoy. Take the chance, make the most of it, because this is one in a lifetime experience. Thank you very much for coming here today.
Thank you so much, Santiago, for such a kind introduction and also for hosting me here. I'm really looking forward to our interactions, and it's definitely an honor to present here to this group. So like Santiago mentioned, today I'll be presenting on our work on algorithms, software, and hardware accelerators for the next wave of genomic data. But before I do that, maybe I'll add to the introduction which Santiago provided. As he mentioned that I started my lab at UC San Diego about 4 years back in 2021, and we span this intersection of algorithms, biology, and computer architecture. And we have this research mission that we would like to develop parallel algorithms, software, and domain-specific hardware accelerators, which enable faster and cheaper progress in biology and medicine. So with that goal in mind, we actually work on a number of parallel computing solutions, which range from developing solutions which map well to HPC clusters or GPU processors, as well as FPGAs and custom chips. So depending on the problem, we choose like what would be the most appropriate solution for that. And hopefully today I will get to span a number of tools that I've developed in, that my lab has worked on in recent years in each of these areas. But the key goal that we are trying to address in our lab is to help answer the central question, which is how do genome sequences, for example, this large collection of genome sequences, how do they really code for this complex life? So genome sequences, as you would all know, is life's instruction manual, and how does it encode these instructions, which help form the complexities of life that we see around us, right? So that's the goal that we are trying to answer. And the reason why we are so excited about this problem is because we think that genomics is really at its inflection point right now. And that's for two reasons. One is that it is really cheap nowadays to collect massive scales of genome sequence data, and that's due to advances that we made in the sequencing technologies, the DNA sequencing technologies. In particular, if you look at this particular graph that NIH routinely shares, you will notice that the cost to sequence a human genome, for example, has dropped dramatically, over 500,000 fold in about 20 or 25 years, which is way faster than Moore's law, right? So what this means is that the data has become really cheap. But more importantly, now it's also easier or relatively cheap to also edit the genome sequences, right? So you can now change the genome sequences in life and in vitro, and you can also study the effect that that change would have on certain function, right? So what this means is that not only is the data cheap, but with computations, we can now make predictions on which part of the genome codes for which function. And with these experiments, we can now validate those, right? So we have sort of completed that feedback loop. And that's why we are living in a very exciting time in this area. And so to that end, next what I will do is try to present what are the specific waves of genomic data, or where we expect the subfields of, of genomics, where we expect large amounts of data to be generated and have massive implications in both scientific research and medicine. So to just name a few, one area that we are very excited about is what is called pangenomics. And here the idea is that we collect large amounts of sequencing data from populations of individual species to gain insights into the genetic diversity, adaptation, and evolution across these populations, as well as to study how the sequences code for or relate to genetic disorders and predisposition to certain diseases. The other area which I'm we have been very excited about or very keenly interested in is that of pathogen genomics. And here the idea is that we sequence genomes of infectious agents such as SARS-CoV-2 to track their transmission patterns, detect outbreaks, monitor antimicrobial resistance, and also guide public health interventions. Then there's this area of comparative genomics, where we are not sequencing individuals in a species, rather we are sequencing individuals across multiple species, right? And there, the hope is that we would be able to identify the conserved and divergent elements in order to understand or uncover the mysteries of evolutionary relationships between these sequences and how the different gene functions are encoded. And finally, there's this area of metagenomics, in which we sequence not individuals, but rather we sequence like the environmental samples, right? So we are sequencing all the DNA content in a particular sample to understand, for example, how microbes would affect the gut health or the fertility of the soil. But also it's being used, this metagenomics is being used for detecting signatures of antimicrobial resistance in, for example, sewage samples these days. And whichever subfield you pick in this broad area of genomics, you will see that there's an exponential rise of data in all of these subfields. And as a result, the tools that we've developed, let's say a decade or two back, really are finding it hard to scale to keep up with this rapid expansion of genomic data. And that's what we are really excited about. And so today, what I'll try to do is to cover at least one tool that we worked on in each of these applications. And one thing that you would notice throughout my presentation is that there would be a particular focus on two fundamental methods that are very common to genome sequence analysis. And those are, number one, the problem of sequence alignment. And here the idea is that you're given a collection of sequences, for example, DNA sequences shown on the left. And what you want to compute is a sequence alignment of those. Here I've shown a multiple sequence alignment of all 8 sequences being aligned together. And the goal here is to find the correspondence of characters, right? So which characters line up, and in this problem, you sort of add gap characters as well, so that each column has a character from each of the sequences, whether it could be the DNA character or a gap character, right? And the goal is to maximize the similarity between the sequences across columns. And the second problem that we deal with is that of phylogenetics. And here the idea is that you want to take these sequences and understand their evolutionary relationships. The idea is that each of these sequences have evolved over time, right? That we are collecting. They are related to each other through evolutionary relationships. So we would like to decipher from the sequence itself what sort of relationships do they hold, right? And typically that relationship is depicted in the form of a tree, although networks are also common. And if time permits, I will also cover some of the works that we've developed in acceleration of these fundamental methods. So the focus is not just on the applications, it's on like fundamental acceleration of these two methods in sequence alignment, in sequence analysis. and hopefully I'll be able to touch upon those as well. But before I move on, I'd just like to acknowledge a bunch of collaborators that have helped us develop many of the tools that we've recently released, spanning these applications. In particular, like Santiago mentioned, I did my postdoc at UC Santa Cruz, and I have a bunch of collaborators active from there, but also various collaborators at UC San Diego and EBI and others. But also we've got we are thankful to our funding sources, which includes the CDC, CDPH, the Hellman Fellowship, and also some of our industry collaborators that includes EMT and Amazon. So with that said, maybe I'll talk about the first application, which Santiago also mentioned, related to Usher, and that's the area of in the area of pathogen genomic surveillance. So let me first introduce the problem that we face in this particular area, in the area of pathogen genomic surveillance. I think you all would be familiar with the picture sort of that I'm showing on the left. The idea here is that you have infectious pathogens like the virus, which is replicating, right? And it's infecting more individuals. As it replicates and infects individuals, it also acquires mutations, which means that it is changing its forms. So that I have depicted by different colors of the pathogen that's shown on the left. But we in reality don't get to see this picture. We don't get to see mutations happening in real time of the virus. What we do get to see is we can sequence an infected individual and collect the genome sequence of the pathogen that has infected that individual, right? So let's say we've sequenced these 6 individuals that have this type of transmission pattern shown on the left, and let's say that these are the genome sequences that we've collected, right? And as you can see that some genome sequences have mutations with respect to the first sequence. Let's say that that's the A, which is at the root on the left. And so while it was spreading, it was acquiring some of these mutations, and there are 3 mutations in this simplistic figure, right? So what we can do with this genome sequence data, and one of the primary goals is that of phylogenetic inference, right? In the rest of surveillance efforts that I will show, this is a core method. And the goal here is to take these genome sequences and sort of infer their evolutionary relationship or reconstruct their family tree-like structure, right? And so what happens in this structure is that the sequences which are more closely related to each other typically group together in the tree. For example, A and C, which are basically the same sequence, grouped together, they are closer to each other than, let's say, F, which has a mutation with respect to the C, right? So as you would see from this figure, it sort of resembles the transmission pattern that I've shown on the left, not exactly, but from this phylogenetic tree structure, we can start inferring the various transmission patterns that could have occurred. So basically, this tree forms the basis of many evolutionary and transmission dynamics study that we do for the pathogen, right? So that's the idea. So the point that I'm trying to make is that this phylogenetic tree construction is like a key step in our surveillance efforts. And one key application of this effort is that of naming the variants of the pathogen. So during the pandemic, I guess many of you might have been familiar that we were naming the different strains of the pathogens like Omicron, Delta, Alpha, and so on. But in scientific literature, they also have a particular nomenclature system, which is scientifically derived. For example, alpha was called B117. And that name is sort of like an address on this phylogenetic tree. So this B117 is actually a sublineage within B11, which is itself a sublineage of B1, which is a sublineage of lineage B. And initially, there were A and B as the 2 lineages, right? So sort of these scientific names are depicting the address in the phylogenetic tree. So they are phylogenetically derived itself, right? And the most popular nomenclature that we use was called Pango, which also has these names like B117 and so on, which was proposed in the initial days of the pandemic. For example, in a bioarchive preprint released in April 2020 and has been widely used for our various scientific discourse, right? And till date, more than 5,000 such lineages have been named, which means that every single day since the beginning of the pandemic, we've been naming, even till today, roughly about 2 lineages per day, actually more than that. So this is very important in keeping track of how the pathogen is evolving. But this system that I just described, the lineage naming system, requires a phylogenetic context. In particular, it needs to know when new sequences come up, how are they related to their neighboring sequences, right? Which are the neighbors that they have in the tree, and also which new mutations have arisen. That's what this system requires. So for example, you could have 4 sequences that I've shown highlighted here in red, which are just placed, and you need to know like what were the neighboring sequences. And in this case, they fall within B11, which is why we would name it B11 point something, right? When we give it a name, if this becomes a new lineage, but also it has a long branch which has several of these mutations on that branch, right? So both of this information is necessary to be known. And this application really works only when you have comprehensive phylogenies constructed. So what I mean is that you want a tree which incorporates all sequences collected worldwide historically, right? If you try to subsample and only use a small collection of those sequences previously collected, you could end up with corrupt naming, and you might also necessitate renaming in some cases, right? So it's very important that we maintain comprehensive trees. And that is the problem that we were trying to solve. But it was very hard during the pandemic because, as you could see, that the number of sequences were growing exponentially. In fact, within a couple of years, SARS-CoV-2 became like the most sequenced species on Earth, with millions of sequences collected. So because of this exponential growth, it was really hard to maintain these phylogenies. In fact, most phylogenetic inference tools of the past, they were based on this assumption that you have static data sets, not growing data sets, static data sets. So you just do a one-time de novo inference of the phylogenetic relationships. And they used to use very computationally intensive methods, which would not scale. In fact, in a single day, you would have more sequences than a tool could handle in like 24 hours period. And so it was almost impossible to incorporate all historically collected sequences with these tools. And naturally, this problem was highlighted in a commentary that was published in Nature. Specifically, it said that if you want to track these variants faster, you have to fix this bioinformatics bottleneck. And it was a huge bottleneck at that time. So our development of Usher was based on a certain key insight. One of those insights was that rather than having static data sets and trying to de novo infer the tree each time from scratch, it would be a lot more efficient if we could leverage the tree that we had inferred on the previous day and just incorporate the new sequences that were collected in the last day or so onto the tree without disrupting the relationships that you had already inferred, right? So that is what we call the online approach, right? So you are incorporating this, sort of like placing new sequences on the tree and optimizing the tree rather than trying to infer everything de novo. It's also based on this key insight that the sequences are in fact very similar. If you look at various strains that I've shown here, sequences of various strains, they only have few mutations between them in their spike regions, just as an example. And as a result, you can apply a lot of approximations to the inference method. And one particular approximation we applied is called maximum parsimony. I won't get into details in the interest of time, but the approach that we ultimately invented made use of this particular data structure, which we'll allude to even in the later half of my talk. The key idea is that you have this tree structure inferred, and on the nodes of the tree, you've already labeled the mutations that you've inferred, right? And when you have new sequence coming up, which has certain extra mutations with respect to the reference sequence, which is stored at the root, what you can do is a brute force search across the tree on how to place that sequence such that it minimizes the parsimony score. That's the criteria, right? So with the fewest additional mutations, you are sort of explaining all sequences that you see on the tree. And in this case, S7 gets placed over here as shown. And once it gets placed, you can also infer the mutation that occurred on its branch easily. And as you can see on the left and the right, that both these data structures are a form of mutation annotated tree. It has a tree and the mutations are annotated on the different branches. So as a result, the output is basically similar to what we had used as the input. So now new sequence comes up, we can incorporate on this structure itself. So this is how we can maintain the tree. In fact, this is what we use to maintain the SARS-CoV-2 phylogeny, which is comprehensive and it's still being maintained on a daily basis. In fact, when we cross the milestone of million sequences, we publish this press release. And we think we were, in fact, the first group to generate a phylogeny using whole genome sequences for any species with million genomes. Usher, because of the way it was, the approximations plus parallel methods that it implemented, it was orders of magnitude faster than techniques for phylogenetic inference that existed back then. But more importantly, because of its speed, it enabled some real-time analysis for this pathogen. In particular, what people could do is just upload their sequences on this web interface that we have for Usher. And within seconds to minutes, they would have those sequences incorporated on the giant phylogeny. And as a result, it became really easy to name and track variants. And in fact, this is the way the Omicron variant, B11529, that's the scientific name of the Omicron variant, was named. These sequences got placed, and on that branch, we had many important mutations that Omicron carried. And within days, the lineage was named, and WHO called it the Omicron variant. That was the speed at which we were operating back then. And in fact, even today now, it has completely changed the game of genomic surveillance. So besides naming of variants, it has led to many other applications like monitoring of different variants or tracking introductions, like how is one strain circulating to some other part of the world and causing a cluster of infections over there, or predicting how the sequence relates to the fitness of the virus. So people develop machine learning models based on our data structure, which takes the genome sequence and predicts how transmissible it is, for example, or predicting what mutations could occur in the future, analyzing super spreader events, and also wastewater surveillance that I'm going to mention in later part of my talk. But one thing that I would want to mention is that the efficiency of Usher was largely dependent on this particular data structure that we created called mutation annotated tree. And it's important to mention the concept here where the important property of this data structure is that we store one root sequence, a sequence at the root of the tree, and we annotate mutations on the tree, right? So the nice property is that you only have to maintain one genome sequence and a set of mutations on the tree, right? The nice thing is that any sequence at the tip of the tree can be easily derived from this data structure, because you can apply the mutations that occur on the path from the root to that tip sequence, and apply those to the root sequence, and you can extract out the tip sequence. So for example, the tip sequence at the top of this tree can be easily derived from the root sequence and applying the four mutations that are shown on the red path highlighted over here, right? So that's the nice property of it. So it's sort of achieving compression. Even if you have a million sequences on the tree, it's the same idea. You have one sequence that you store and just a bunch of mutations on the tree. And many of these mutations are shared by multiple sequences. So there's a lot of compression that you can achieve as a result of this, which makes it very, very efficient. In fact, as you can see, if you had raw sequences being stored in a file for like 6 million of those, for SARS-CoV-2, it would take 190 gigabytes, and you get 2 to 3 orders of magnitude improvement in storage efficiency by using mutation annotated tree. So this, of course, was very effective, and we then published a lot of related papers in various journals, which includes like how we maintain the tree, but also we did the largest recombination study, which we published in Nature. How do we optimize the tree, and so on and so forth. So yeah, that's one topic that we've been very interested in working on, and now Usher has been expanded to many other pathogens, and we continue our efforts in this direction as well. So with that said, maybe I'll move on to the next topic that I'm going to cover, which is related to metagenomics and also very, very related to pathogen surveillance that I mentioned earlier. So before that, let me just maybe introduce very quickly what sort of application within metagenomics are we talking about, and that's that of wastewater-based epidemiology, or WBE in short. So here the idea is very simple. It's like environmental sequencing, where we are taking sewage data, also called wastewater data, and the idea is that we apply genome sequencing techniques to it to detect genomic traces of the pathogens which are infecting a community from which that sewage data has been collected, right? And the idea is that infected individuals are constantly shedding the genome sequences of the pathogen, and that gets collected in this in the sewage water, and you can just directly sequence it to get an estimate of what infectious diseases might be circulating in that community, right? And there are many reasons why this is advantageous over doing clinical sequencing, which means that someone gets infected, goes to a clinic, and develops symptoms and goes to a clinic and then gets sequenced, and that gets deposited on a public database. This has more advantages to that. One is that it's more cost effective, right? Rather than sequencing every individual who has been infected, you just sequence like a whole population through just one sample, right? That's the idea. It's less biased because you don't need to have access to healthcare facility, or you don't need to develop symptoms in order for your the sequence of your pathogen to be sequenced, right? And also it's more timely because you don't have to wait for people to develop symptoms, go to the clinic, and then carry out the sequences and so on. So it actually saves time, and people have shown all of this. during the pandemic that it actually helps in faster monitoring of different pathogens, this particular technique. And because it was so effective, it has been now widely deployed across the world. In fact, there are more than 50 countries across the world which are actively using wastewater-based epidemiology, not just for SARS-CoV-2, for many other pathogens as well. But many of these efforts did begin during the pandemic time. And many of these countries are from low and middle income countries where, clinical sequences tends to be very expensive as well compared to WBE. Another trend that we are seeing is now that the whole world has declared that the pandemic is basically over, the clinical sequencing efforts have also dramatically dropped. So this is showing you like how many sequences we're collecting in different months since the beginning of the pandemic to roughly end of 2023. And now it's actually even lower, but you can see that there's been a rapid decline in the later half of 2023. And as a result, what we are seeing now is that it's become harder to discover new variants. There are longer delays when we find new variants, but also we are now coping with this fact that we are getting lots of inaccurate and incomplete information about how the pathogen is evolving just because we are not sequencing enough clinically. So one question that we've been asking in my lab is, can we use wastewater epidemiology to, cover the gap that is being created in clinical sequencing efforts, right? And many of these press articles are also talking about something similar, trying to highlight how wastewater epidemiology can be used for pathogen tracking. So there are existing tools which do wastewater-based epidemiology, but the downside of these tools or the limitation of these tools is that they are mostly focused on estimating lineage abundances or viral activity. So what I mean is lineage abundances is actually more detailed. I'm showing you a plot over here, which basically shows you what these tools are analyzing. Essentially, what they do is they collect samples at different points, and they are trying or different time points, and they are sort of estimating which variants are circulating in what proportion, right? So lineage level information, like is it B117 or XBB, which kind of variant is circulating and in what proportion? And that's basically what they're trying to do with the wastewater data. And the way they model this is actually pretty simple. It's a variant of this technique that I'm showing you here, Freya, which is like considered the state of the art in this domain. Essentially, they are applying like a deconvolution algorithm where they are trying to solve this equation. The idea is very simple, that you have an observation B, which basically shows you which mutations that you find in the wastewater that you've sequenced, right? And then there's this matrix A, which is basically the lineage barcode information, which is derived from Usher's mat. Essentially, what it's saying is that for every lineage like B117, XBB, or whatever lineage you care about, you basically annotate in this matrix, a 0-1 matrix, which mutation it has, right? That's the matrix it has, right? 0-1 information. Does it carry this mutation or not? That particular lineage, right? And so what you're trying to do is solve for this X term. It's a vector of abundances of different lineages, such that you are minimizing this term ax minus b, such that the abundance sums up to 1. Abundance of all lineage sums up to 1, and each abundance is greater than 0. That's the constraints, right? And if you solve for this equation, you basically get x, which is the frequencies of different lineages that you see. That's the idea. As soon as you derive this, you can actually generate plots like this at different time points, right? And this particular tool was published in 2022 in Nature and has been widely used since, but it has some downsides, right? One particular downside or limitation is that it is only trying to estimate the sample at the resolution of lineages, right? So whether it's XBB or B117 and so on. But in reality, we often see that there's a huge genetic diversity within lineages as well. So just to give you an example, I'm showing you a BA1, which is one of the lineages that we see in Omicron. Two sequences which were collected at roughly the same time points from the same region had about 50 mutations separating them within the same lineage. So these previous tools would only tell you that BA1 circulates at 50% abundance, but we would not know whether it's just one cluster or two separate clusters separated by several mutations that are circulating, right? So it won't tell you that information. The other limitation is that they can be sometimes inaccurate too, especially when you're talking about really deep lineages. What I mean by deep lineages is that lineage which itself has a lot of genetic diversity within it, then the chances are that it will be inaccurate. And this is just showing you like the root mean square error of that, and it's actually high in some cases. The other limitation is that this technique, by definition, it requires that you have already it only estimates abundances for lineages you've already designated, right? But as I told you that now we are encountering more delays in even designating lineages that we have sequenced, right? In fact, this is showing you some data for only the recombinant lineages, but it's true for most other lineages as well, that it's common to see designation times spanning from weeks to months for sequences that you've just collected, right? So because you collect the sequences, then you that has delay, but then you also submit it, and then there are people who curate it, and then it gets officially named. Once it gets officially named, only then can you estimate its abundances. Till that point, you are basically not even treating it as a new lineage. You are treating it as one of the existing lineages that are circulating. So it has timeliness issues as well because of this. So we took this approach of using the phylogeny that we are already maintaining to sort of improve the wastewater epidemiology. So I'll get into the details of this, but the key idea is that we take the mat that we are already maintaining and updating on a daily basis for Usher, take the wastewater sequencing reads, and then apply this technique, which we call WEP, to it, and not only estimate the lineage abundances for the lineages that you've designated, but also identify which haplotypes in the mat are most likely contained in the wastewater sample, right? So these are the haplotypes which we have on the mat, and mat has all sequences that you've collected. So haplotype is just like a genome sequence that you've collected before. Which of these best explain the sample that you've sequenced just now? But in addition to this, also list down the unaccounted mutations. So these are the mutations that are present in the wastewater sample, but cannot be explained by the haplotypes that you've selected. So they were not previously seen clinically, right? So or they cannot be explained by just clinical sequences. So they might be signs of new variant emerging, for example. And finally, we also do this higher resolution read to haplotype mapping. So you are taking every single read and identifying among the haplotypes that you've selected, and there are 3 of them selected here, H1, H2, H3. What is the proportion of each haplotype? And which read maps to which haplotype, right? And in most cases, you will see some reads map to multiple haplotypes that you've selected, right? That's kind of the high level idea. But essentially what we are trying to do is improve the resolution of the wastewater analysis. So previously what people do would sort of look like the image on the left, where you cannot really appreciate the fine features. But with WEP, you are able to do that, right? So you get a much more detailed and cleaner picture of what's going on in the wastewater sample rather than just doing a high level lineage analysis, right? So that's the idea. So I'll quickly move over the algorithmic overview. Essentially, what we are doing is we are using the Usher's mat, and we are taking the sequencing reads coming from the wastewater and doing a phylogenetic placement of those reads onto the mat. So the idea here is that you're not placing the whole sequence like we did with Usher, but we are modifying Usher so that you place a subsequence, right, a sequencing read, and find its most parsimonious location onto the tree, right? So the fewest additional mutations explain its position on the tree. And once you place all these reads, you can just look at, for example, the distribution of how these placements map to select certain haplotypes which are present in high proportions, right? Just as an example. It's a little bit more involved than that, but that's essentially what we do. And then what we can do is just add a few neighbors to it just because we want to give the next algorithm more choices, and let the deconvolution algorithm that you earlier had choose among those haplotypes which ones are most likely, right? So essentially, phylogenetic placement is doing a pre-filter and selecting a small number of haplotypes which the deconvolution algorithm can handle. And then the deconvolution algorithm gives you the proportion of each haplotype that is in circulation. And then you can do a more refinement of this to get the final estimates. But at the end of this, you will get haplotype proportion. So the haplotypes that you've selected, what frequencies do they occur? And in this case, it's 50, 40, and 10. Which lineages are in circulation, and that's easy to derive from the haplotype proportions because you know which haplotype belongs to which lineage. A set of unaccounted mutations. So these are the mutations which the haplotypes don't carry, but we see in the wastewater sample. So it can help you track the new variants which might have emerged and also assign read to haplotypes. That's kind of the idea. So we released a preprint on this work recently. And what we find is that with this approach, the WEP's approach, you actually dramatically improve the lineage abundance accuracy as well. So you can see that WEP's RMSE, and lower is better in this case, is way better than other 2 approaches, which are based on the deconvolution like algorithms which I mentioned, or EM algorithm. But more importantly, it can also improve the resolution of the analysis. So in this case, I'm showing you a simulated example where you had multiple lineages circulating, co-circulating at the same time. But in EG5, we actually simulated a sample similar to what we actually saw in San Diego, where we had haplotypes of multiple haplotypes, which are shown in these circles onto the tree circulating. And what star is showing you is which haplotype web selected to be present in the sample. And as you can see, there's a strong overlap of the haplotypes which were circulating in the sample and the one which web actually estimated. In fact, in most cases, we found that web estimated the haplotype within a mutation distance of the true haplotypes. And so what you can see in EG5 that there were actually two distinct clusters, one red cluster and one green cluster, and we can now identify those. So when a public health official looks at this, they can tell that EG5 is not one, like it's not like one type of EG5 circulating, but there are actually two distinct clusters that are co-circulating at the same time in a given county or region, right? We can also use it to track introductions. Now, this is an application which earlier we only used to do with clinical sequencing data. But now, because we can identify nodes in the tree and their proportions, let's say you did this, you collected this sample in Delaware, and you found that these two nodes were selected with proportions 4 and 1%. What you will see is that the nodes surrounding the one which Webb chose are all from California. You can see it from the names that they are all from California. So when a public health official would look at this, they would generally conclude that there's a cluster that has now emerged in Delaware, and that cluster must have its roots from California. So it might have been introduced from some traveler, for example, who must have come from California and infected an individual in Delaware, and then that has spread, that infection has spread and caused a cluster to occur in Delaware. So these are the type of applications that you can now do with wastewater, with which earlier would only be possible with clinical sequencing data. We also analyzed several real-world samples which were collected across the world and shared online, and found that web results generally correlate better with clinical sequencing data than prior tools. So with that, I'll end this part of the talk and maybe move on to the new application of pangenomics. But before I do that, maybe I'll see if there's any question. Pause for a minute and see if there's questions.
Any questions so far, also from the online audience? It's a tricky time of the year. Some people are in conferences abroad, so we can take any questions. Speak up. Well, they can use the chat if they want to put any questions. I have a question in meanwhile. I would like to understand the size of these matrix operations that you're putting there, because as far as I understand, the size of the linear algebra operation you're doing there is quite big.
Yeah. So that's why these approaches didn't scale to millions of sequences, right? So if you think about this matrix, right?
Yes.
This matrix.
That one.
So it has all lineages
Yes.
and all mutations that can occur. So like I said, currently the number of lineages for SARS-CoV-2 is roughly 5,000.
Yes.
Right. So that's 5,000 rows. And mutations can be in the order of, let's say, few tens of thousands. So there are 30,000 positions in SARS-CoV-2.
Yes.
Not all of them you would see, in any sample, but let's say 10,000 or so. So it's actually 5,000 across 10,000, very rough approximation, right? Matrix that you see. So it's several millions already.
Yeah.
And this method doesn't scale if one of the rows becomes, let's say, 1 million.
Yeah.
It won't scale, right? So that's why it's very important that you have fewer rows for A, right? So that's what Web is doing. It's taking 16 million initial sequences, doing phylogenetic placement and reducing the search space down to, let's say, a few thousand, which this type of deconvolution method can handle. But rather than looking at only the roots of the lineages, it's actually going within the genetic diversity of lineages to select haplotypes.
I see.
Right? And that's where the higher accuracy comes from.
I see. Because otherwise it's not.
Otherwise, this root might be too distant from what's actually there. Right? As I showed you that it could be even 50 mutations apart.
Yeah.
But 10 is very common, for example, and in that case it can get confused, like where do those 10 come from, and so it can try to account it from some other lineage, the abundance. So that's the benefit of phylogenetic placement.
Very nice. Any other questions from the audience? I was distracted.
Yeah, I have one. Here you are saying that you map the reads directly to the Azure tree. In this case, which type of reads are you using? Because I can imagine that these reads have to be very high quality in order to not introduce much error, right?
Yeah, that's a very good question. So during the initial placement, we actually use literally every single read. Now, some reads may have errors and they can cause misplacements or whatever, right? But bulk of them will get placed very close, and it's only a initial filter in some ways to the final step. Then in this deconvolution step, before that, we actually apply some additional filters where we discard some low frequency, which could be erroneous and things like that, right? So there are some optimization, but placement we do for every single read. Initial placement we do for every single read, and then there is before this lineage-based estimation that I mentioned, this step we would do some post filtering as well, like quality control.
Okay. I don't know if the question is naive, but what type of technologies are you working with?
So yeah, we actually showed results for both long reads and short reads.
Okay.
So bulk of wastewater is short reads.
Short reads Illumina-like.
Yeah, but long is also not that uncommon. Actually, people use nanopore, for example, MinION and so on.
Nanopore?
Yeah.
Okay.
There's a lot of nanopore data out there too.
The whole sequence molecule, or they chop it and they
yeah, so
by
wastewater
region?
will always be chopped. So generally, you don't even get 10,000 long reads.
Okay.
With the wastewater, you would get maybe a few hundred, 500, 1,000.
Okay.
Because in the sewage itself, it gets fragmented, right? The genome gets fragmented. So you almost never end up seeing. That's why this problem is difficult. Because from short fragments, you're sort of trying to reconstruct the whole picture. And it's also mixtures of different pathogens. So it's a fairly hard problem to crack.
And how about the quality of those, out of curiosity, how about the quality of observed for those sequenced samples? Is good, is bad? Have you?
So it's similar to, so error rates are similar to, what we generally encounter with clinical. So it's the same technology. So the sequencing instrument would have roughly the same error rate, like 5-10% or whatever, right? The difficulty is actually in the fact that you have mixtures, right? Where you have many lineages or many haplotypes co-circulating at the same time. Just think about like a city like Barcelona. If let's say there's only one wastewater plant,
yes,
so many people are infected with so many different variants, and now all of them have fragments within them, and you are trying to reconstruct the whole picture with it. It's not going to be easy.
Right.
To solve, right? So that's the real challenge with wastewater epidemiology, how to take that mixture and deconvolve it. And I don't think the error rate also poses a challenge, but not as much as like dealing with a mixture,
right?
Complex mixture. Yeah.
Right. Good.
Okay.
Thank you.
So with that, in the interest of time, maybe I'll move on and talk about pangenomics, which is another area which we are really excited about. Like I mentioned earlier, the idea is that you collect sequences of genomes from a single species, and this can have many applications. For example, studying how genomic variation leads to diseases in humans, or some types of desirable traits in plants and animals, or antimicrobial resistance, for example, in different pathogens. So if you look at current pangenomic analysis, right, and there are many works on this, it's a rapidly emerging field, you will see that they are generally limited to tens or hundreds of genome sequences at a time, right? And I've cited a few of them in this example. And the question that we are asking in my lab is, how do we scale our pangenomic analysis to handle millions of genomes, right? So that's going to be the focus of my talk. That's what we are looking at this point, like just how we did with SARS-CoV-2, can we actually scale to millions of sequences, and lots of applications that we will enable as a result of it. But currently, if you look at the pangenome data representation, which is a key aspect of pangenomic analysis, how do you represent pangenomes, right? The collection of genomes. You will see that most popular representation is focused on these graph-based techniques, right? So the idea here is very simple, that you have these different genomes and their variations have been highlighted in colored boxes. What you would do is the common regions where subsegments which are shared between all sequences, you would share them as a common node, and that node corresponds to a sequence. When they diverge, for example, you will have divergent edges in the graph leading to two different nodes carrying two different sequences. And then they can converge back when they have a common sequence again, and so on, right? So the idea is any sequence that you have in the collection can have a unique path in this graph, which can be used to describe that sequence, and as a result, you can store the collection. Now, this type of representation tends to be excellent when you want to solve the problem of reference bias, which means that earlier analysis was based on a single reference for that species, and that used to introduce certain biases in both read alignment and variant calling. If you replace them by graphs, it improves, right? The alignment improves, the variation calling improves, all of that improves. But in other types of analysis, I think there are certain limitations that these graphs pose. For example, you would notice that these graphs don't have any evolutionary information between the sequences being stored, right? So you have many sequences, but how are they related to each other? Which sequences more similar? Which sequences are more similar, which are more dissimilar, right? You are not storing that information at all. You can infer it from the graph, but you don't explicitly store that information, right? And that can be very valuable if you do for downstream analysis, like the one which I mentioned in earlier talks, in earlier part of the talk. There's also no mutational information being stored, only variation is being stored. What I mean is you are encoding the differences between sequences in their different paths, but you're not encoding what changes, specific changes, and the sequence of those changes that led to those differences, right? So mutation has a subtle difference from variation. Variation is the outcome of mutations, right? Mutations are the events that happen, and variation is the net outcome of that. So don't store that. They're also hard to visualize. As you can see, this graph, it's actually very few sequences, but it's very complicated for end users to actually visualize and make sense of such graphs in some cases. But because we are also from a computer architecture background, we know that if you want to accelerate these type of graph-based analysis, they tend to be very hard. Any graph-based algorithms tend to be hard because they have irregular parallelism, complicated memory dependencies, and so on. So we generally tend to prefer regular linear-like structures more than graphs because they tend to complicate that accelerator design too much. And also they are storage-wise not very efficient, as I would show in later part of the talk. So this is, I think some of these limitations are well known, and as a result, pangenome representations, there have been two different directions of work which people have generally taken. One direction is more about how do we compress it, because the graph-based representations are not very efficient. So people have worked on formats which take a collection and just compress it. So it's only about storing raw sequences in compressed form. So think about it as an alternative to gzip, right? And that's what these tools are. AGC and Minify are basically those. They are more complicated versions of gzip, essentially domain specialized versions of gzip, which give better compression rates. And the other focus has been on representing more information. So an idea that has been proposed recently is of pangraph, where you store the evolutionary information of different sequences, so for phylogenetic structure within them, or ideas proposed by tree sequences, where you are trying to encode for recombination and other type of mutations. But naturally, they would have trade-offs. What can represent more versus the memory efficiency of it? So generally, you will see that the methods which have high memory efficiency, like Minify and AGC, tend to represent less information. They are only focused on compression rather than storing mutation, variation, or phylogenetic information, and so on. The ones which store that information tend to be low on memory efficiency, right? Like graph-based representations. What we are trying to do is to reach a better trade-off point where you have higher representative power, in fact, higher than the current approaches do. It stores more information, but memory efficiency is comparable or better than the best compression techniques, which are only meant for raw sequences. It's a very hard goal to achieve, but I'll show you that we do end up achieving that. And the key idea is the same that we use for Usher, that of evolutionary compression. So you remember the property I told you that collection of sequences which are evolutionary related, the way you can store them is have one sequence at the root, a phylogeny on which mutations are annotated, and any sequence at the tip of the phylogeny can be derived easily by just traversing the path to the root, applying the mutations to that root sequence, and you get it, right? You get the final sequence. But in Usher, we had some limitations in our MAT structure. One is that it was reference-based. So essentially what we did was every mutation that we computed was with respect to the sequence at the root, right? So it had this reference bias, which is what we are trying to get rid of in pangenomics. We also only stored substitutions. We did not store indels, and that is very complicated because if you have insertions and deletions, they actually modify the coordinate system. So if you see the mutations here are based on some coordinates, they are reference-based. Now you have to change that in order to incorporate indels in the pangenome representations. But indels are important in pangenomic analysis. In fact, they're important for SARS-CoV-2 as well. And finally, we were only restricted in our phylogeny to tree topologies, right? In fact, if you look at even SARS-CoV-2, it is mostly a tree-like structure, but sometimes it actually recombines, which means it forms hybrids. And those hybrids can be derived from like two sequences. So they can have more like a network-like structure. This is showing you an example of SARS-CoV-2 network. So we would like to get rid of all these limitations. And that's exactly what my three students, Sumit, Harsh, and Yusiang have worked on. they applied evolutionary compression to pangenomic applications. Essentially, the idea is that you take the alignment and phylogeny and sort of represent them through this evolutionary compression like structure. In the interest of time, I won't get into too much detail. It's actually the coordinate system, we replaced it by the multiple sequence alignment, sort of what I showed you before. Its columns define the coordinates. And you treat gaps as a special character. So besides ACGT, you can also have gaps. In fact, you can also have ambiguous characters like N's and X's and so on, which can represent multinucleotide ambiguities. And then you have some methods that we incorporate to have like large insertions and deletions being stored, so that you can have like blocks which can mutate. And instead of having just one tree, we have a network where you can have some sequences which can recombine to form the root of another tree, right? And to store that, we can apply complex mutations which only have breakpoint information in them, which regions cuts off and recombines to form another child sequence, for example, right? So that's basically what we did. But the key result is that we have more representative power. It's actually the first format which combines alignment, phylogeny, and mutations all in a single format, as far as we know. So what this means is it's not only information rich, but as I will show later, it's also compact and very scalable. In fact, we applied this on few thousand genomes of different pathogens. Because the reason why we are restricted to the scale is because the alignment techniques are not scaling. So we are also working on that. But at this scale also, you see that the compression rates are really good. Compared to GFA, 19 to 680-fold, VG, 6 to 152-fold, GBZ, which is a more compressed version of VG, again, 5 to 42-fold, and so on and so forth. In fact, also what we see is that it scales when we have more sequences, scales well when we have more sequences. So what that means is, when we had 20 sequences, you may see a 4-fold improvement in compression, but when we move to 20,000, it goes to 150-fold. So what I'm trying to say is, if you have more sequences, the chances are that the gap would widen between PanMan and other compression formats. In fact, we use this format to actually build the first pangenome of all 8 million sequences that are publicly available for SARS-CoV-2. And it only required 366 megabytes of information. Now, think about it. If you had a multiple sequence alignment for it, it would be in terabytes, a few terabytes. So it's about 3,000-fold more compressed than MSA in its raw FASTA form. So I've skipped some other data just in the interest of time, but we have actually evaluated on lots of data sets and including with AGC and Minify, and we've seen high compression rates with respect to them as well. Now, the cool thing that you can do with this method is not just extract out sequences, so it's not just compression and decompression, but you can extract out meaningful information as well. So for example, you can extract out the graphs. You can extract out the GFA because you have the variation information also stored implicitly because you've stored mutations, and from mutations you can derive variations. You can extract out VCS, you can extract out the whole multiple sequence alignment, phylogeny, all of it. So this is what makes it really powerful. And now we are actually working on expanding this to more complex genomes like human genomes, because they recombine more frequently. You don't have a single parent, you actually have two parents, right? So there's a lot of recombination going on between those two. They also have multiple chromosomes and so on. Polyploidy, which means you have two copies, one from each parent. So how do we extend this format to that and also compress it effectively? That's what we're working on, and GPU acceleration of construction. So with that, maybe I'll skip this particular application, the comparative genomics application, and just tell you a little bit, maybe it's just like take 5 minutes to explain like hardware acceleration work that we are working on. So the key idea is that, as you would have seen, that alignment and phylogeny keeps recurring in a lot of the analysis that we do for genome analysis. And of course, this is a problem that we are interested in from a very fundamental level as well. How do we accelerate this more broadly? So if you look at, for example, the problem of pairwise sequence alignment, where you have two sequences, let's say R and Q, reference and query, we generally solve them using 2D dynamic programming. I'm sure many of you are already familiar with it. You take these two sequences, arrange them in the rows and columns of a dynamic programming matrix, compute the dynamic programming scores, and from the optimal path of the from the matrix, you can actually derive the alignment, right? And the problem of aligning long sequences is now becoming very important. So for example, aligning whole sequences of SARS-CoV-2 is important, or aligning long read sequences to the reference genome, also very important, right? This problem is important, but traditionally these are very hard to accelerate in hardware, because when you have long sequences, you have more states in the dynamic programming matrix to store. And so it becomes nearly impossible to have full matrix computations being done. Both computationally it's hard, but also from a memory point of view, it's very hard. And as a result, developing accelerators can also be hard. In practice, people don't use full matrix computations generally. They tend to use banded alignments. What that means is they only compute a small search space within the dynamic programming matrix, and that is what generally gets used. Now, my previous work, which Santiago did mention about Darwin, it introduced a tiling strategy, which can be used for computing these long alignments by dividing them into sub-alignments which have fixed size in memory. And that is great for hardware acceleration, because in small amount of memory, you can actually get an accelerator implemented, which means you can even parallelize it, and which means you don't have to depend on off-chip memory for acceleration. So that worked great, but it was a heuristic in place. And what we've recently done with our work on Talco is develop a method which can do optimal tiling with banding, right? That's the idea. And it's based on this notion that many of these dynamic programming methods have this property of convergence of traceback pointers, which we can leverage. And that's how we developed this method called Talco, which is optimal under those constraints, and was actually published in HPC last year and also won the best paper nomination for it. And recently what we've done is we've expanded that to multiple sequence alignment problem. And in fact, my student, Yuxiang, and also Sumit, they worked on GPU acceleration of multiple sequence alignment problem, which applies tiling approach to profile alignment steps. And what we found is that it actually performs not just better in terms of runtime, it's actually the fastest among these techniques, but also has better accuracy because of some of the algorithmic modifications that we did. This work, by the way, will appear in ISMB next month. But this is the method that we use for generating MSA of 8 million SARS-CoV-2 genomes. Now this is a multiple sequence alignment, which means that it's not doing whole genome alignment, so it doesn't account for rearrangement. But in SARS-CoV-2, the good thing is we've not seen rearrangement, so it actually does, it's equivalent to a whole genome aligner as well. And it required 28 hours on 2 GPUs to compute this whole genome alignment, which is actually the largest multiple sequence alignment as far as we know till date. And now we support AMD GPUs as well, in addition to NVIDIA, and support protein alignments on this framework too. And there's also some work that we've done on using high-level synthesis for accelerating dynamic programming. The idea being that many dynamic bioinformatic algorithms that you see use 2D dynamic programming, just variations of it for many different applications. And what we also see is that generally they map really well to systolic arrays, just like how matrix multiplication maps well to 2D systolic arrays. Any 2D dynamic programming algorithm maps really well to linear systolic arrays. And you can see lots of papers recently published, which basically use the same idea. So what we asked in our lab is, can we use high-level synthesis to add a layer of abstraction in a high-level synthesis workflow, so that you can have a front end on which you can specify this algorithm in C++ API. And the idea is that you don't have to use any HLS or digital design background to do this, but you also have a backend which has all the HLS optimizations already in place, so that it can map that algorithm that you specified onto a systolic array very efficiently. In fact, we did end up successfully implementing this framework, and we implemented like 15 different dynamic programming kernels, all the way from protein aligners to Viterbi algorithms, Smith-Waterman. All of this we implemented recently, and found that compared to software baselines, we actually get high speed ups on FPGA, real FPGA implementations on AWS F1. And also when we compare to HLS baselines, we get improvement compared to, for example, Vitess HLS, because of more optimizations in place in our backend. And if you compare to handwritten RTL baselines, we are nearly equivalent. Not as good as a Verilog code that you would write from scratch, tailored one, but nearly as well. So, but in terms of design productivity, it's a huge improvement. What would take weeks to months can be reduced down to hours to days with this framework, right? So it's a good way to explore dynamic programming algorithms on this framework. So with that, the talk has basically come to an end, but I'll just quickly summarize through these points, the different tools that we worked on, different applications as well. But yeah, thanks a lot for listening, and I'll be happy to take any questions.
Thanks to you, Yatish. Be careful, guys, because this gentleman here is going to leave us out of job if So we'll be happy to take any questions from the audience online in the chat or here at the auditorium. Any questions? Anybody?
I have a question about the GPUs you used for doing the multiple sequence alignment. It was like these high-end GPUs, like H100 or
It was A6000.
okay. Because you think it makes sense to use like consumer grade GPUs, because for the high-end GPUs you pay a big premium for all the tensor cores and high memory bandwidth, which here I don't know, but probably you don't use them much.
Yeah. So we use a desktop class GPU for that analysis. So it's not the super expensive high-end GPU. And also Ampere, so it's pre-Hopper GPUs, and there are more advanced versions. We do have DPX support, by the way. So the optimizations which are more are on modern GPUs. What we found is actually GPU acceleration we get about 2 to 4-fold speed up. Then so Twilight also has a CPU version, the multiple sequence alignment tool that I talked about. It also has a CPU version, and that's also really fast. It's about 4 to 8 folds slower than the GPU, but it's still pretty fast. Because of the tiling that we developed, right? The tiling strategy, it actually works really well on both CPUs and GPUs. It actually gets huge benefits even there. So it actually gets even orders of magnitude speed up on CPUs itself. I don't know if I fully answered your question, but
yeah. Well, just
Okay.
curiosity, which GPUs were you using?
yeah.
Regarding the DPX that you mentioned right now, what is your experience regarding performance?
That's a great question. So because we also do traceback, right? We also do traceback, and DPX doesn't have argmax, right? We don't see much improvements in very initial empirical evaluation that we've done. It would have really helped if they had that argmax also incorporated, but it gives like minor improvements, like maybe 20%, 30% of that order in the end-to-end speed ups, mostly because we also have to then compute the traceback pointer, right? If you
So 20% is
maybe 20%, yeah, around that much, yeah.
More questions? More questions here? I have a more general question about the algorithmic approach that Twilight acquires as to tackle the multiple sequence alignment. Multiple sequence alignment is one of those problems that, there are books and books and books and algorithm, different approaches. I'm very curious. I don't know if this is a very general question, and probably we don't have much time here, but I would like to just sneak peek into what is the algorithmic approach you chose for the Twilight.
Yeah. So sorry for glossing over that, but in the interest of time, I just wanted to highlight the work rather than get into many technical details. But there are multiple optimizations here in place. But a very broad idea is that it's a profile-based aligner. Okay. So generally in profile alignment techniques, what you do is it's a greedy approach in a way where you have sub-alignments and then you align sub-alignments, right? So the multiple sequence alignment problem behaves like a 2D dynamic programming pairwise alignment problem, but the pair is now sub-alignments, right? So how do you decide the order in which you align sequences? That's decided by the guide tree, right? So that's the phylogenetic relationships between these sequences. So you first have to construct this guide tree. That's provided as input to Twilight, right? And then basically you're going from tip to the root, right, of the tree and aligning sequences. So that's the idea shown here, that you have this 3 sequences that you're aligning. So you combine 1 plus 3 first, right? And that sub-alignment you combine with 2 over here. That's the idea. Now, this problem of this profile alignment has been used in cluster, muscle, and those kind of techniques. So it's very similar to those. We use this guide tree also in a very similar fashion. Now, the key optimizations that we performed is, first of all, on GPUs, you exploit parallelism on 2D dynamic programming, but also we used the styling approach into Talco. And then we had one step in which we found better accuracy, which you mentioned, which is to do gap removal and gap additions later. So what I mean by that is some columns in the multiple sequence alignment have too many gaps, like 90% gaps, 95, 98% gaps, right? What we do is we actually remove them out before we do a pairwise alignment and then just add them back as introducing new gaps in the second alignment, right? So that gives better accuracy results. That's why we've seen with multiple evolutionary simulation results. The other thing that you have to be careful about when you are doing GPU acceleration is when you are aligning 8 million sequences, like I mentioned, they can take hundreds of gigabytes of memory. You don't have those on a GPU, right? So you can't even store the raw sequences in their form. So we had to do a number of optimizations in place in order to optimize for handling of the memory. So what we do is we only transfer sequences which are, for example, aligning in a sub-alignment. Next, what we do is we transfer the profiles on the GPU, which are easier to store than exact sequences, and CPU maintains the actual alignment within the profile. This is an example, right? And because of tiling too, we get benefits of lower memory usage on the GPU. So number of optimizations in place. The manuscript will come out soon about it, which actually has a whole section on how we optimize for memory constraints within Twilight.
We should read that. Definitely, we need to read that. And if I can elaborate, I understand, it is to my understanding that underneath each of these progressive sub-alignments, you're using the Talco approach, converging, and you're applying there okay, that's very neat.
Yeah,
very
so,
nice.
so this gives huge benefits on the GPU. So actually our traceback state is maintained only on shared memory of the GPU.
Right.
That
is the key.
Yeah, that's the key. And as soon as you do that, you get huge speedups on the profile alignment step. And that's basically the bottleneck in most software, the profile alignment step. The other optimizations that we had to do in Twilight are mostly related to memory. How do we, because you have fixed amount of memory on a GPU, how do we optimize for that? So there's constant back and forth. And we also have lots of file storage in place. So sometimes even CPU may not have enough memory. So we actually store the sub-alignments in files, then read it out, and then sort of concatenate all the final alignments in place. So yeah, from the traceback path, you can actually reconstruct from sub-alignments the newer sub-alignments. Yeah.
That's really cool. That's really cool. Folks, if you want to tackle a big problem, this is the one. So keep an eye on this. Any more questions? I may? No?
Yes, in a slide, I think I've seen dynamic time warping.
Yeah. So that was DPHLS. Yes.
Yeah, what type of heuristics are you using for it?
So you mean this one, right? So right. So
Implemented for it, yeah, right? An implementation for it.
Yeah, we have an implementation
Yeah,
for
what's
this. So,
like strategy are you using? Or
so by strategy, so we are basically, so to just give you a very high level idea, we are implementing something very similar to what Squiggle did. So there the idea is that you store sequences in complex numbers. And then they already specify the dynamic programming or dynamic time warping equation in place. It's actually more simpler in this case than even Smith-Waterman, right? So we just applied the same dynamic. So we just code the same equations that they have on our framework. And that's what we implemented and then compared to it. So the outputs match exactly to Squiggle. Yeah, so I don't know if I fully answer your question.
I just wanted to know if you used any tiling or...
Oh, tiling, yeah. So I should mention that. So this is not, so here we evaluated on shorter sequences, but DPHLS also, if you apply tiling to it, it supports tiling. So we've demonstrated that in DPHLS, you can implement Darwin-like tiling strategies on top of it. So it can work with long sequences too in a similar form. But this was evaluated mostly on shorter sequences, this particular framework. But yeah, you can apply tiling to it.
Good. Well, I think we can thank Yatish for being here in this very nice talk and give him another applause.
Thank you so much.
Thank you, folks online. Thanks for